Hello everyone. Welcome to today's live broadcast, Applying the MinION, Microbial Genomics and Complex Metagenomics. I'm Brenda Kelly Kim of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Oxford Nanopore Technologies. Oxford Nanopore is developing and commercializing a new generation of nanopore-based electronic systems for analysis of single molecules, including DNA, RNA, and proteins. The handheld MinION device is designed to provide simplicity of workflows and real-time data streaming. For more information, please visit www.nanoporetech.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit your questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to get to as many questions as time allows. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button, lower left. I would now like to introduce today's speaker, Dr. Daniel Turner. Dr. Turner is the Applications Director at Oxford Nanopore Technologies, where he is responsible for developing library preparation methods to allow MinION users to work with a broad range of sample types and working with external collaborators to demonstrate new uses for the technology. I'll now hand it over to Dr. Turner for his presentation.
Great. Thanks very much, Brenda, for the introduction. And thanks everybody for tuning in as well. As Brenda said, my name is Dan Turner, and I'm Senior Director of Applications at Oxford Nanopore Technologies. I'm assuming that some people watching this presentation will know a fair amount about Oxford Nanopore and the technology, but also that some people won't know a thing. So I'm going to spend the first few minutes talking about what the technology is and how it works. Okay, this slide illustrates Oxford Nanopore's vision for a complete end-to-end solution from sample to analysed results. So we're not just a company who makes a sequencer. We want to provide the whole end-to-end process from the sample, whether that's blood, leaf, cell culture, or whatever, through to sample extraction, library preparation, sequencing, to full analysis of the results. So returning the biological information that the experiment was set out to investigate. The pane on the right shows our PromethION, which is a scalable, high-throughput, standalone device, and that's going to be available towards the end of the year. But for this presentation, I'm going to be focusing on the MinION, which is a lower-throughput, portable device. This uses the same sensing chemistry as the PromethION, so everything that's possible on the MinION will also be possible on the PromethION when this is available. So the concept of using nanopores to sequence DNA has been around for a couple of decades. And it's illustrated here in a sketch from David Dooner's notebook. In essence, you have a nanopore sitting in a membrane of some sort, and you have an ionic buffer at either side. When an electrical potential is applied, a current flows, so ions move from one side of the membrane to the other. And as an intact DNA strand passes through the pore, the current changes. This is more or less how we do it today, as I'll explain shortly. It looks simple, but the challenge is in making it robust, scalable, and high throughput. Now, there's been a lot of academic research into various uses of nanopores as sensors over the last 20 years. For instance, detecting small molecules such as TNT, or using them to sequence proteins. And it's this versatility that lies behind Oxford Nanopore's preference to be described as making sensing devices rather than just making DNA sequences. What is a nanopore, after all, other than a tiny hole? And as long as you can get the molecule of interest to interact with the pore in a characteristic way, then it's possible to analyse it. However, for this presentation, I'll be focusing on the use of nanopores to sequence DNA and RNA. Okay, so how does it work? You may have come across the use of solid-state nanopores in various contexts, and though we have strong interest here, the MinION device currently uses protein nanopores. These have some distinct advantages for us, namely that in a batch of protein nanopores, all of them are more or less identical, so they behave in the same way. And we can modify them using conventional molecular biological techniques to give us particular properties. Our nanopores sit in a membrane, not a lipid membrane because that isn't robust enough, but we use a synthetic polymer, which has got very high electrical resistance. We add a salty buffer solution to both sides of the membrane, and we apply an electrical potential. The current can only flow through the pores, and we measure the changes in the current as analytes pass through the pore. So the picture now shows the open pore current when there's nothing blocking the pore. Small molecules, as the figure illustrates, choke off the current to some degree, and larger molecules choke off the current even more. This isn't quite how things work when we start to look at DNA, but it does illustrate the general principle. When we come to look at DNA, the bases are joined together, and in the method which we call strand sequencing, DNA molecules pass through the pore like this, intact. When the DNA passes through the narrowest part of the pore, it chokes off the current. Several bases can fit into the narrow bit of the pore at any one instant, so we're actually measuring the current from a short word, rather than from an individual base. If 5 bases fit into the narrow part of the pore, then we're reading 5 bases, and we have 4 to the power of 5 different combinations because of the 4 different types of base. So that's 1024, which means that we have to fit 1024 different current levels into the detection range. But because we make the DNA move through the pore 1 base at a time, every time we move, 4 letters in the narrow bit of the pore stay the same from one word to the next. And this means that when we take the next current reading, there are only 4 possibilities for the identity of that new word, or the identity of that new base, word. Over the course of the entire strand, we generate a series of current levels, each of which is related to the previous one by 4 of the 5 bases, and we use a hidden Markov model to convert this signal to base calls. Okay, so there are 3 essential components in nanopore sequencing. There's what we call the motor protein, there's the nanopore itself, and there's the membrane in which the nanopore sits. We have a variety of different nanopores which we can use, and each interacts with the sequencing template molecules in a slightly different way, meaning that the electrical currents are different, and we can use this property to our advantage. As mentioned earlier, we need a membrane with high electrical resistance, so the current can only flow through the pore, not across the membrane. Lipid bilays are not very robust, and although we did use them at one stage, we now use a synthetic polymer which is even robust to blood. And finally, we have the motor protein. In the previous slide, we talked about the need to move the DNA through the pore 1 base at a time, so that we can work out what the sequence is. So we need a way of doing that. If we just let DNA or RNA go through the pore unhindered, it goes through something like 1 million bases a second. And that's a lot faster than the frequency at which we measure the current, so we need to slow it down. We do this by using an enzyme which we call a motor protein. The motor protein actually serves a few functions. It slows down the rate at which the DNA or RNA passes through the pore. It acts as a ratchet, meaning that it moves 1 base at a time, pausing briefly each time it moves, which allows us to measure the current. And it's also able to unwind the duplex ahead of it. So rather than needing to make our sequencing template single-stranded, we can use double-stranded templates. This makes library preparation a lot easier, as we'll see in the next slide. Library preparation is the process of converting extracted genomic DNA, RNA, or amplicons into a format that's compatible with the sequencing system. This is shown here for genomic DNA. Oxford Nanopore's current genomic DNA library prep is quick and straightforward to perform, and it's capable of generating very long fragments of DNA, which will give long reads. The user can shear their DNA at the start if they like, but it's not a prerequisite, and it's clearly not what you would want to do if you wanted very long reads, but it does make sense for some applications. After shearing, or not shearing as the case may be, the fragment ends are repaired, and a single adenosine is added, and then adapters are ligated on. Now, one of the adapters that we use is a hairpin, and this connects both strands in a duplex, and it allows a strand to be sequenced followed by its reverse complement, all as part of the same sequencing read. This means that we get 2 looks at each region of DNA, and it allows us to sequence with greater accuracy. At the opposite end of the hairpin, we have a forked adapter. And this other adapter has various features which increase the efficiency of the sequencing reaction. So it has a low secondary structure, meaning that the strands are more easily captured by the pore. It has a motor protein preloaded onto it, meaning that all ligated strands will pass through the pore at the right speed. And it has a region to which we hybridize a tether oligonucleotide, which increases the sensitivity several thousand-fold. This tether oligo has a hydrophobic group on one end, which means that the strands are able to associate with the hydrophobic membrane better. The adapter also has a feature which stops the motor protein working its way along the strand until the strand is captured by the pore. So in solution, the motor protein sits on the strand, not moving, but as soon as it is captured by the pore, then that allows it to start processing along the strand. The library prep looks similar to other next-generation sequencing library preps, but there are a couple of important differences. Firstly, there's no PCR. Although it's possible to do a PCR as part of the library process, for instance, if you don't have a lot of DNA to begin with, PCR introduces bias. It reduces the fragment length, and hence the read length, and it adds time to the library preparation process. So if you can avoid PCR, it's best to avoid it. To the best of my knowledge, we have the only sequencing technology that's capable of sequencing the actual strand of DNA that came from the organism of interest. And this means that we can get extremely long reads, tens to hundreds of kilobases long so far, and we can detect modified bases directly. The second difference in the library prep compared to conventional next-generation library preps is that we don't routinely perform size selection as part of it. The reason we don't need to do this is because nanopores randomly and constantly sample strands from solution. So as soon as one strand has passed through the pore, another one gets captured. And even if there are shorter strands present in a library alongside longer ones, the shorter strands take less time to pass through, so they take up proportionately less sequencing time than longer strands. Not having a requirement for size selection means that we avoid a laborious step, and although it's one that can be automated, the equipment needed to do this is large and expensive. That said, if you want a lot of reads that are 100 kilobases or more, size selection becomes more important, because DNA of this size is fragile, and so preparations of this size often contain high proportions of much smaller fragments of DNA. And as the slide shows, we also have a barcoding kit available for people who are, for instance, looking at smaller templates such as viral genomes or apricons, and they want to pull and sequence many of these together to get better value from the sequencing run. Hopefully it's clear from before, but it's worth clarifying that the motor protein is not attached to the pore. If it was attached to the pore, then if something happened to the motor protein, such as it denaturing, then we'd lose that pore. So the motor is attached to the template strands, and as a strand is captured by the pore, it starts to move along the strand, unwinding the helix ahead of it. The unwound strands thread through the pore one base at a time, and because of the hairpin, both strands of the duplex go through. Data from both strands is combined during the analysis into what we call a 2D read. This has got higher accuracy compared to either strand alone. Once the template is finished translocating, the motor protein diffuses away, and the pore is then free to capture the next strand. We load templates into the flow cell at a concentration that's sufficient to minimize the open pore time between one strand and the next. It's typically a fraction of a second, so the pores are occupied pretty much all of the time. And a final word about the motor protein is that we can adjust the speed at which it processes along the template strand by increasing or decreasing the concentration of cofactors. And this means that we can make the system run a lot faster and we can generate more data. In our so-called slow mode, we process the DNA at about 30 bases per second per pore. But in fast mode, we can get about 500 bases per second or even more per pore. We've talked a lot about DNA, but it's also worth mentioning that nanopores are capable of sequencing RNA too. This is a work in progress for us because we've been focusing mostly on DNA, but we have been getting very good results so far with RNA directly. It's typical in sequencing to convert RNA to cDNA to complementary DNA before sequencing. And this is something that we can do easily on the MinION, but it might not always be ideal because reverse transcriptases don't always process to the end of transcripts. And it's also typical to do a PCR with cDNA libraries, and that's got all the disadvantages that PCR brings, as discussed earlier. But also during the conversion of RNA to cDNA, information about modifications of the RNA bases is lost. We can go about sequencing RNA directly in several ways. Unfortunately, all of them work. We can ligate adapters straight onto the RNA strands, and we can pass those through the pore. We can prepare a first strand cDNA, and we can link this to the RNA strand so that we can sequence the RNA followed by the cDNA, or vice versa. This gives us the advantage of seeing what the polymerase thought about the RNA bases, which provides us with more information about modifications. As the data in the bottom right shows, we see a change in the current levels as a modified base passes through the pore. And it doesn't just affect one current level. As you'll remember, we're looking at short words, and a modified base will appear in several of these as it passes through. So we get several looks at it, which means that we can be more confident about it. Here's a look at the MinION. And as you can see, it's small, so it's portable, and this opens up the possibility of using it outside of a laboratory environment. We've already mentioned that it's capable of producing long reads. As far as we can tell, the only thing which limits the read length is the quality of the input DNA. One thing that I haven't covered so far is the real-time nature of the data generation. Strands pass through the nanopores intact, and the current levels are measured as this happens. It means that as soon as a strand has started to pass through a pore, the data then becomes available for analysis. The other factor here is that there's not a fixed runtime with a MinION, or a PromethION for that matter. And together, these are things which open up a lot of possibilities, such as being able to stop a sequencing run as soon as you have enough data to answer the question that you set out to answer. Or to put it another way, you can leave the device running until you see the thing that you want to see. But also it means that you can perform something akin to sequence capture, except that it's on the device. So you sequence the first few bases of a strand, and if it's not from the region or the organism of interest, you can reject that strand by reversing the electrical potential on that individual pore. The MinION connects to a laptop via USB, and the USB connection provides the power for the MinION, and it's how the data gets transferred from the device. In the bottom left panel, you can see the MinION in more detail. This is actually a more recent version of the device than the one on the top right, and the main visible difference is that the lid's hinged rather than separate. All of the sequencing reactions take place on the flow cell, which is the consumable. This contains the pores and the membranes where the library is loaded. Underneath the flow cell, we have a custom application-specific integrated circuit, or ASIC. And this is what controls the applied potential, and this is how the electrical currents are measured. The rest of the device consists of various bits of electronics and fans, and a fan to keep everything cool. In the bottom right pane, you can see a closer view of the area of the flow cell where the pores and membranes are. Each chip has about 500 single nanopores. So that describes the hardware and the wet lab side of the system. It's also worth mentioning briefly the METRICHOR platform. This currently hosts a range of core applications built by Oxford Nanopore, with the ultimate aim of supporting a variety of applications built by third parties, essentially an app store, some of which will work with the raw sequencing data and some of which will be higher level apps built to aggregate metadata over time, for instance, or for groups of users. In the not too distant future, it may target cell quantifiers or integrations with the Internet of Things. So having heard all about the technology, if you're interested in trying it out, we're running what we term MAP, which is the MinION Access Program. That means that for an entry fee of $1,000, you'll get a MinION and a supply of consumables and analysis software, which allows you to run your own experiments. There's a very strong and supportive online community where you're able to discuss ideas, to get tips and find collaborators, and you're also able to buy further consumables through our online shop. For details of that, please visit our website, which is www.nanoporetech.com. We also have a booth at this conference, a virtual booth, so please do check that out as well. Okay, so onto some microbial applications of the technology. The first application I'd like to talk about is de novo assembly of microbial genomes. Using short read sequences to try and assemble a genome has been likened to taking a novel, putting it through a shredder, then trying to piece it back together again so that it can be read. This may be more easily visualized as being like a jigsaw, or maybe rather 30 copies of the same jigsaw, only with each one cut differently, and then with all of the pieces mixed together. Short reads here would be equivalent to having very, very small jigsaw pieces. And to find out where the two pieces fit together, it requires a lot of searching. It makes for a computational challenge, and the results tend to be imperfect. It's particularly difficult to assemble genomes that contain repetitive regions, for example. If your pieces are longer, let's say your reads are longer, let's say 5 kilobases rather than 100 bases, then the task becomes a lot easier, and you gain the ability to resolve repeats that are a few kilobases in length. As the read length goes up to, for example, 50 kilobases, the job's even easier. We're not yet at the point where we can generate reads of 500 kilobases, but there's no reason why that would not be possible from the pore perspective. It's really just the integrity of the DNA. So as mentioned earlier, the challenge becomes one of trying to extract and manipulate DNA strands in such a way that the strands don't break. Okay, so we wanted to test our ability to assemble a genome using only nanopore reads. We wanted to start off with something small, but something that was more interesting than lambdaphage. So we got some DNA from a collaborator, Chantal Abidjan in Marseille, and the DNA comes from quite an interesting source. They discovered a virus in Siberian permafrost. It had been there for more than 30,000 years. The virus is enormous by viral standards. It's about 1.5 micrometers long, and this virus infects amoebae. It's possible to resurrect the virus in the laboratory. Now, as the authors point out in their paper, as permafrost thawing out, the ability of viruses to come back to life after tens of thousands of years could have some quite worrying ramifications. But that's not what we were looking at. We were interested in trying to assemble the genome, which, as it turned out, was fairly AT-rich. We had to PCR the library, which was unfortunate because it meant bias and shorter reads, but we didn't have access to much DNA, so we had no choice. But with a bit of help from Nick Lohman, well, actually quite a lot of help as he did it entirely, we were able to assemble the genome into a single consig. That's one fragment covering the whole genome. As you can see from the number plot of the assembly against the published reference, things match up well. I don't have any alignment metrics for this one, but I don't think they'd have been especially good, since this was done before Nick and Jared Simpson had finished their polishing tool, NanoPolish. But this brings us nicely to the next slide. So earlier this year, a landmark paper came out in Nature Methods by Nick Lohman, Josh Quick and Jared Simpson, in which they demonstrated that it's possible using only MinION reads to assemble a bacterial genome into a single consig. The first part of the method works by taking nanopore reads, detecting overlaps between them, and then subjecting the overlap reads to one or more rounds of error correction, and then using just the information contained in the reads themselves and no external sources. After this step, the corrected reads are then assembled using the Celera assembler. It's possible to stop the assembly at this point, and the authors found that when they did this, they got something like 98.4% identity compared to the finished reference genome for the K12 strain of E. coli. But they then developed an additional step, which doesn't work in base space, but it uses a processed form of the electrical current levels generated by the MinION device, which we call squiggles, and this increases the identity to about 99.5%. We were really excited by this, and we wanted to try it out for ourselves. We've been able to assemble the data set released by the authors of the paper, but we wanted to look at a genome where we had produced the sequencing data for ourselves. So we made a couple of libraries from the E. coli strain SCS110, which we just happen to have in our -80-degree freezer because we use that as competence cells. One of the libraries that we made was a standard library, which had been made by fragmenting genomic DNA, as described on the earlier slide. And the fragment length distribution was centered at around 6 kilobases. The other library was one prepared in almost exactly the same way, except that we omitted the fragmentation step. So the library fragments represented the original fragment length distribution much more closely. And the other difference was that we took more care with this library not to shear it during library preparation. So no vortexing, for instance. The read length distribution of this library was centered on around 30 kilobases, but with a tail of very long fragments going to well over 100 kilobases. And the longest read that we got from that library was 192 kilobases. I know the fact that we look at both strands of the DNA duplex confuses matters here, but what I'm really saying is that we sequenced that strand for 384 kilobases, and we then combined the data from the two strands into a single 192 kilobase read. We wanted to see what the effect of having these long reads would be on assembly, so we took enough of our 6 kilobase data to cover the SCS110 genome to 40-fold, and we ran that through, not nanocorrect this time, because we hadn't managed to install it in-house, but through a piece of software called PBCR, which also error corrects reads and feeds them into the Celera assembler. We got several countries after assembly, as you can see in the number plots, where we used the closely related K12 strain as the reference. The M50 was about 3.8 megabases, and we got around 98% identity. It's worth mentioning that the error correction of nanocorrect seemed to do a slightly better job on our data than PBCR, and we didn't do a polishing step here, which explains why the identity is slightly lower here than it was with the published assembly. We then repeated the assembly, but with a mixture of the 6 kilobase reads and some ultra-long reads, very roughly 10-fold each, so 20-fold coverage overall. After putting these through the Celera assembly, you can see that we get a single contig assembly, which might not be all that surprising, but it's nice to see. The M50 here was about 4.5 megabases, which is the entire genome. The percentage identity was very slightly lower here because the coverage is lower. But anyway, enough about assembly for the moment. The bottom line is that long reads make de novo assembly easy. Next, I'd like to talk about a real-time application for species identification that we like to call What's in My Pot, or WIMP for short. As I mentioned earlier, as soon as a strand of DNA has passed through a pore, it can be base called and then it can be analysed. In the WIMP workflow, species are identified and classified in real time. The classification relies on a data structure which maps all k-mers of length 24 present in the reference database to note on the NCBI taxonomy tree. Thanks to this pre-processing, new reads can then be quickly classified by looking at k-mers, rather than by aligning them against the original reference. WIMP uses Kraken to map all k-mers to calculate the least common ancestor and to filter to determine the most likely placement in the taxonomy tree. The output is a taxon ID for which a classification score is computed. The higher the score, the more confident we can be that the classification is correct, and then you get a blue colour on the taxonomy tree, and it's yellow if it's less confident. The user can set confidence thresholds depending upon whether the aim is to achieve maximum specificity or maximum sensitivity. And this is an application that's available through METRICHOR. So we recently expanded the database to include viruses as well as bacteria, and we're currently expanding this to include fungi as well. We tested the viral database by buying a range of different viral RNAs from a commercial supplier, preparing libraries, sequencing them, and putting the data through the workflow. As you can see, we included Pithovius in there as well, and although Pithovius is a DNA virus, so it needs to be prepared in a different way from RNA viruses, it's one of our favourites, so we included it in the mix as well to make a rather bizarre metagenomic sample. We then run the WIMP workflow, and we obtain the taxonomic tree shown here on the left. Now, if you remember from the previous slides, the colour codes, you'll hopefully remember that blue is high confidence and yellow is low confidence. So you can see from the tree that the viruses were identified by WIMP with high confidence. Now, as well as running the libraries through WIMP, we also had barcoded each library so that we could look at how well the WIMP read counts corresponded to the actual reads that we got, i.e. does the WIMP workflow introduce bias in any way? So the bar chart that's just come up shows that the two sets of reads, two sets of data agree very closely, indicating that the WIMP workflow is quantitative. But we wanted to look at that a bit more closely because it's potentially a very important feature. So to do this, we took a selection of bacterial genomes for which we had a good quality reference genome, and we prepared libraries from these, and we pulled them in arbitrary ratios. We then used quantitative PCR to quantify the relative abundance of each genome in the library pool, with the intention of comparing the qPCR quantities to the WIMP quantities. We had to convert the qPCR data to make this possible, because the qPCR result is a measure of the number of copies of the genome, whereas with whole genome sequencing, the number of fragments and reads is proportional to the size of the genome. As you'll see from the plot, the qPCR counts agree well with the WIMP data, indicating that WIMP, including the sequencing and all of the other parts of the process, is quantitative. We're often asked how sensitive this workflow is, i.e. down to what level can we detect viruses and bacteria in a sample. Now, given that the workflow is quantitative, it's essentially a question of how many reads you get from your sequencing run. And this is just a matter of how long you leave the device to generate data. So if you're interested in detecting very low abundance species using the WIMP workflow, then it's just a matter of leaving the sequencing running for longer. As I said earlier, because of the real-time nature of sequencing on MinION, you're not confined to a fixed runtime. So if you need more data, you just leave it running. So to summarize, WIMP's a quantitative workflow for real-time species identification. All right, let's see how this works in a real-life situation. As I'll talk about in a few slides' time, the limitation of the WIMP workflow is that the sequence of the species to be identified needs to be available and in the database. If WIMP encounters a species that's not in the database, it will be able to place it further up the phylogenetic tree at a position where it's more confident. Anyway, there'll be a bit more on that later. We wanted to apply WIMP to a sample where it would recognise most of the various species in it. So we thought of looking at cow's milk. This is a good sample because drinking unpasteurised cow's milk is something of a fad. And apparently it tastes amazing, but there's a downside to it. And that downside is that the whole reason that the milk gets pasteurised in the first place, which is that raw milk is replete with pathogenic bacteria. As the table at the top shows, it's not uncommon at all for people to become ill from drinking more cow's milk. Well, that's not massively surprising, given that cows spend so much of their time with their udders dangling in their own excrement. The table below lists a few of the pathogens commonly found in unpasteurised milk, and there's some really nasty things in there, such as Listeria, E. coli, 0157, and so on. So we wanted to get some more milk and see what was in it. But just in case we got a sample of milk that was comparatively pathogen-free, we thought it would be good to get some Listeria DNA and to add that in for good measure. Now, because of the health risks involved in drinking unpasteurised milk, it's not as easy to come by as the average molecular biologist would like it to be. In fact, in New York State, where we did these experiments, unpasteurised milk is barely legal. It's not legal to buy it at retail, but it is available if you go direct to the farm. But who goes to a farm to buy their milk? Well, exactly. So there are various people who will sell raw milk off the back of a lorry. And because of this, raw milk has acquired the sobriquet moonshine. We got this from a smuggler who you can see in the top left picture. Obviously, her identity is protected to stop her getting into trouble with the law. And we got a few different samples. So we got cow's milk, we got goat's milk, and we got sheep's milk. Just to increase our chances of having something interesting to detect, we left the milk samples at room temperature for a couple of weeks before extracting the DNA. And we then extracted the DNA, spiking the Listeria into the cow's milk at the earliest opportunity in the reaction. We left the sheep's and goat's milk as they were, with no added pathogen DNA. Now, when we run the sheep's and goat's milk through the WIMP workflow, we identified a range of bacteria with differing levels of confidence. This is what you expect from running WIMP on what's effectively an environmental sample. If we look at the tree at the top, you can see calls down to the subspecies level, but with different degrees of confidence. So, for instance, and I'm going to try and annotate this because it's maybe a little bit difficult with the size of the picture, but we see a strain of Serratia liquefaciens with very high confidence. Remember that blue is high confidence and yellow is low, which indicates that we're seeing this exact strain in the milk. A little further down, we see, hopefully I'm circling the right ones, we see 3 different subspecies of Lactococcus lactis, all at low confidence. But the node that's one level higher, i.e. Lactococcus lactis, the species, is at much higher confidence. And this indicates that we do have Lactococcus lactis in the sample. No big surprise there. But we just don't have one of the 3 subspecies that are present in the database. And it's a similar story for the sheep's milk as well, as shown in the bottom pane. None of the things that we found in these milk samples will cause too much surprise. They're all things that are either commonly grown in milk or they're common in a farm environment. When we move on to cow's milk, we see a similar story in many respects, in that a lot of the bacterial species present in the milk are ones that you would predict, so Lactococcus and Pseudomonas. But what's especially interesting here is that a good number of the species that we identified are ones that are known to cause mastitis. Now, mastitis in dairy cows is particularly bad because it leads to necrosis of other tissue, as shown in the photograph. The milk that we got presumably came from several cows, and because of the way we got the sample, we can't exactly trace it back to specific animals. But what we do know is that mastitis affects up to about 50% of dairy cattle, so it's pretty much a certainty that this was a genuine detection of the disease. What we also see in the animation on the right is that we were able to detect the spiked in Listeria DNA with very, very high confidence. And the number of counts of Listeria reads was more or less exactly the same as the amount that we added by mass, as you would expect from a quantitative workflow. Okay, so this is an illustration of the sorts of things to which a MinION and the associated workflows can be put. And it highlights the non-lab applications. I mean, you can see how useful this workflow in combination with a portable sequencer would be in monitoring the health and well-being of dairy animals, for instance. But the remaining problem is that to get the DNA extracted and prepared for sequencing on a MinION, you would still have to take various bits of lab equipment, such as a centrifuge, onto the farm or wherever you're working. So to address this need, we're working on a device which we call Voltrax. And this is essentially a MinION-sized sample and library preparation device. The device is going to be compatible with all of the library preps that we have, including cDNA, PCR-based preps, RNA, all of them. And it's entirely powered by USB, making the whole process truly portable. Ultimately, we'll be tuning the device so that it's capable of extracting the DNA or RNA from a range of samples, as well as performing the library preparation at the user's choice. So the user just has to add a bit of sample in liquid form and then sit back and wait, and the sequencer will take care of itself. But anyway, back onto species identification. When we were first thinking about the What's in My Pot workflow, we set out to test it on an environmental sample. And because much of this work was being done in our New York lab, it made sense to try a local sample. We decided to look at some water from the Hudson River to see what was in it. And on the left, you can see David Dye from the New York team, who collected the sample on the New Jersey side of the river. Rather than going for the actual water, in which we thought the bacterial count might be a little bit thin, we went for some of the sludge at the bottom of the river, as you can see in the other photographs. We used a soil DNA extraction kit to get the DNA out of the sample, and we ran it through the WIMP workflow. You can see from the WIMP tree that a range of different bacteria came up, but with low confidence, as indicated by the yellow circles. Excuse me. If we look at the table of species that were identified, we can see that a good number of these are in fact very, very likely to be present in the sample, such as Shionella and Desulfobacter, just not the species and subspecies that are present in the database. But only something like 10% of the reads matched anything in the database. The remaining 90% of the reads couldn't be classified. This indicates that there are a lot of species in the sample that are not in the database because they've not yet been sequenced. So what can we do about that? Well, we'd read a few articles about a device called an iChip. As the sketch on the left shows, the iChip consists of a microscope-sized array of small wells, something like 350 of them. The idea is that you dip it into the sample of interest at such a dilution that you get, on average, 1 bacterial cell per well, and you immobilize the cells in a gel such as agar or agarose. You then assemble the other bits of the iChip, including permeable membranes, which allow nutrients to diffuse in and out, but which keep the bacteria where they are. You then put the iChip back into the environment from which you took the sample in the first place. During this time, the bacteria may or may not adapt to life in agarose. I can't be sure about that, and I didn't really worry too much about it when I was setting up the experiment, as we'll see. The photographs on the right show the iChip in action. We chose to use the stream at the back of the Oxford Nanopore building in Oxford as our sample, primarily because it sits at the confluence of the Littlemore Brook and the affluents from the sewage treatment farm, which makes it a charming place to work. But we're also confident that we could find some bacteria in there. It's also interesting to look here because the stream is a popular site for people to go fishing in. We wanted to know exactly what they were eating. In fact, you can probably see in the central picture on the right, a person in turquoise who's just taken a break from fishing to watch this odd fellow collecting water from the river, which is me. So we immersed the iChip in low melting point agros and then dipped it back into the water, assembled the device, and then put it back into the stream for 2 weeks while I was on holiday, letting the chip do the work. After that, I recovered the iChip, removed all the leeches and slime and other disgusting things. Using sterile cocktail sticks, I pushed the agros plugs into separate wells of deep well plates, which was filled with low salt LB medium, except that the medium had been made with river water, sterile river water, instead of distilled water, just in case the bacteria present had specific nutritional requirements. The plate was then left at room temperature and exposed to daylight for about 3 days. We hadn't taken too much care to make sure that the water sample was dilute enough to give 1 bacterium per well on the iChip, so we included an extra step in the process, which was, after the cultures had grown, to streak them out onto agar plates, also made with river water, as shown in the top right photo. We then picked single colonies and grew them in the same way as before. As the photo of glycerol stocks in the bottom right shows, we've got a range of bacteria, giving cultures in a variety of colours from white to cream to red to fluorescent yellow even. We then extracted the DNA from a few of these using the QIAGEN 500 tip and sequenced them. Excuse me. When the sequences were run through the WIMP workflow, the results were fairly inconclusive. As I said earlier, if a bacterial subspecies is not in the database, then WIMP cannot identify it with any degree of confidence, which explains why the trees on the right show low confidence values, and why there are several hits even though each species was cloned. What we can be confident about is that the species we're looking at belongs to that genus or that family or higher up, even if the subspecies is low confidence. This is illustrated really well by the lower right-hand pane, where we have a Pseudomonas species, which may be Pseudomonas fluorescens, but the confidence is not that high. But it's certainly in the genus Pseudomonas. If we look at the table on the left, we can see that Pseudomonas species are quite abundant in this sample. I guess this method is not dissimilar to digital PCR, where the number of times you see the same thing will indicate how abundant it was in the initial sample. So it's not especially surprising that Pseudomonas comes out as the most abundant. And we've seen a range of things in there, some of which we can identify down to species level, some of which we aren't really sure about yet, but we're going to sequence them and find out. But we thought it would be really interesting to take a closer look at one of these species. Excuse me. The one we chose by random was the one which WIMP said was, what's in my pot workflow, said was a strain of Klebsiella oxytosa. We sequenced this using a PCR-free library with a fragment length of approximately 20 kilobases. We generated about 180 megabases of data, which is about 30-fold coverage of the oxytosa genome. After sequencing, we assembled using nanocorrect because we've managed to get it installed in-house now. And we obtained a single contig assembly covering approximately 6 megabases, which is the typical size of an oxytosa genome. We then performed a whole genome blast on NCBI to find out where this particular species lies in the taxonomy. And as shown on the left-hand pane, by this measure, it's most closely related to the top part of the tree. The closest match was to a strain called KONIH1, which had been obtained from a perirectal swab. And if you remember that our sample came from sewage effluent, it's not all that surprising that it is very closely related. We then made a mummer plot of the assembly against the KONIH1 strain. And you can see on the right that the two are very similar, just with a few bits missing here and there. It doesn't make a lot of sense quoting comparative metrics here, given that the assembly is not the same strain as the reference. But the N50 was about 5.7 megabases, which is the full length of the genome. So we're near to the end of this presentation, and I think it's worth summarising a few things here. That the MinION is a small, inexpensive, portable sensing device, which is capable of producing long reads of sequence information in real time. This lends itself particularly well to easy and inexpensive identification and discovery of microbial species. And when coupled with analyses that also work in real time, it becomes ideally suited to the real-time detection and characterization of microbial species, which we think will be perfect for things like infection control and monitoring food hygiene and safety. So what next? Well, Oxford Nanopore is an ambitious company, and our ambitions extend beyond the confines of this planet. As you might have seen in the press, there are plans to send the MinION up into the International Space Station in early 2016, which will make it the first DNA sequencer in space. Initially, the idea is to see whether or not it's possible to sequence in space samples that have been prepared on Earth. But the longer-term plan is to use the MinION to monitor astronaut health, for instance by monitoring the intestinal microbiome, and ultimately, who knows, detecting extraterrestrial life. Though admittedly, there might be one or two library issues to deal with here if the genomes are not DNA. The pores themselves should be able to handle whatever strands we throw at them. As you might imagine, though, trying to sequence DNA in zero gravity presents a few problems. And this is one of the many places where Voltrax could come in. Because with Voltrax, it'll just be necessary to pipette or syringe a raw sample into the device, and Voltrax will take care of everything else. But in fact, there are far more things to consider when you're trying to sequence in space, as this slide shows, and each of these steps needs to be tested thoroughly before launch. Anyway, that brings me to the end of the presentation. It just reminds me to thank a few people without whose help I would really have struggled here. So I'd like to thank all the staff at Oxford Nanopore, but particularly the applications group, and in particularly Cecil, whose team in New York performed many of the experiments I've talked about in this presentation, and also who helped to grade all of the slides. I'd like to thank Ant Jones, who made the iChip, and also Fernando and Amber, who are the METRICHOR people. But I'd also like to give a special thanks to the gangster rap bioinformatician Nick Logan and his posse, Jared and Josh Quick, and also to Chantal for the gift of the Pithovius DNA. And thanks to everybody for listening as well. I'd be very happy to take any questions.
Thank you, Dr. Turner, for that informative presentation. Before we get started on all of our questions and our answers, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. And we'll try to answer as many questions as we can. Now, our first question. You mentioned that WIMP is quantitative. Does that mean that all MinION sequencing is quantitative?
Sequencing is quantitative. It depends how you go about setting up the sequencing reaction, I suppose. I wouldn't claim that anything that has an amplification step in it could be remotely quantitative. What I would be confident saying, I mean, but it's bioinformatics that we need to do in proper detail so we can give a proper considered opinion, especially for transcriptomics. But I think that without the amplification step, we come much more closer to having a quantitative system. Before I can say yes, it is fully quantitative, we need to validate that. But I don't think there's any reason to suspect that it is not quantitative. We've not noticed any biases, GC biases or AT biases. And like I say, without amplification, that's removing another source of bias.
Thank you. We do have another question. Our audience member asks, I noticed that in the moonshine results from WIMP, reads from cow also appeared, but you said the WIMP workflow was for bacteria or virus only. Does that mean I can customize the WIMP database?
Yeah, very, very, very well observed. Yes, it does mean that you, well, okay, I'm going to have to phrase this very carefully. It is potentially possible to customize the WIMP database. It is not something that we've currently released as one of the workflows. So if you use the WIMP workflow today, it will come with canned workflows for canned databases for bacteria, for viruses, and in the very, very near future, fungi. As to whether you would want to customize it, and it's something that we do debate quite a lot. But I mean, if you know the species that you want to put in the database, then you might be better off not using the WIMP workflow anyway. Its strength is in looking for species where you don't know what you're looking at. So for the moment, you can't customize it yourself, but it is potentially possible.
Great, interesting. Another audience member says, this technology is amazing. Can anyone get into the MinION Access program now?
All you have to do is go to our website, which is www.nanoporetech.com, and register your interest, and then we will be back in touch with you. Like I said on the slide, there's an access fee of $1,000, which means that you get a MinION, you get a supply of flow cells, and you get some consumables. If you want more than that, then you can buy them through the shop. But yeah, the MinION Access program is open to anybody.
Great. We have another question. You mentioned that you can sequence modified bases in DNA and showed it for RNA. Does that mean all modifications?
Well, you'll appreciate that the number of modifications that are out there is vast, and in RNA particularly, it's a frightening number of modifications. So, for instance, for every base in RNA, you might have, let's say, 5 different methylated variants at least. And it's probably the same for, well, not necessarily the same for DNA, but certainly you have, a large number of methylated Cs beyond just the 5 nuclei. What I can say is that when a strand passes through the nanopore, we see it. It's more the question is how we can make sense of it. And so the easiest thing, and what I showed in the slide, is that if you have a modified strand and an unmodified strand, and you overlay the squiggles from those, you can see a difference. And that tells you that there is something there that is not matching with the base call model that we're using. Now, if we knew what the base should have been in that position, and we then see that we've got a deviation from that, it implies that there is a modification there. And particularly if you're looking at an RNA strand and a cDNA copy, so you get to see what the polymerase thought as well as what the pool thought, and you can then try and infer from that the presence of the RNA base, the modified base. So we can tell from, I'd say, pretty much any base that there is something there that is a modified version of the base, and whether to get to the point though where we can put in any strand, de novo sequence it, and call whichever modifications happen to be there. That's going to take us a lot longer because it means that we have to see each modified base in the context that we are going to see it in the real sample. We have to know what we're looking at. Well, first of all, we have to know what to look for. We then have to have strands to train our base caller with that strand in, so we can learn what each one looks like. But in time, yes, we should be able to look at modified and unmodified bases without any prior knowledge or prior assumptions, just by putting the DNA in and working out what it is. But that's going to be some time from now. Getting a message to go to the phone. Right?